All right, I don't think that she needs to hit record. Oh, she does. Okay, great. I will start. Hello, everybody. Welcome to the IBANGS seminar series. I want to encourage anyone who's not already an IBANGS member to become one, because it's a great society and a great conference. As for today's speaker, it's my singular pleasure to introduce Dr. David Jentsch, who will be kicking off this semester's series. Dr. Jentsch is the Empire Innovation Professor of Psychology at Binghamton University, where he maintains a very well-funded behavioral neuroscience research program investigating the genetic and neural bases of the response to alcohol and other drugs such as cocaine, mostly using mice. His work is supported by a number of grants, UO1 grants, P50 Center grants, R1 grants from both NIAAA and NIDA. And on top of that, he's director of Binghamton's NIAAA T32 training grant. And on top of all that, it's the first week of court classes at Binghamton, and he is chairing search committees. He's got his hands in a lot of different committees. Before his time at Binghamton, Dr. Jentsch got his bachelor's from Johns Hopkins in behavioral biology in 1992, and his PhD in 1999 from Yale, where he worked with his advisor, Robert Roth. After a postdoc at the University of Pittsburgh, he started his own lab at UCLA in 2001, where he was until coming to Binghamton in 2015. His work's been very influential to the field of behavioral neuroscience. He's well recognized for his work on animal models of impulsivity, especially in the context of disorders such as schizophrenia and drug addiction. The work he published with Robert Roth and Jane Taylor 20 years ago is still very influential. And since then, he's been capitalizing on his background in behavioral genetics to continue this work. I won't give any details about that, though, because he's going to talk about some of that today. His consistent productivity over the years has been accompanied by several awards, which I won't enumerate now in the interest of time. Just to say on a personal note, David's a great colleague and a great person. I'm sure many of you know that already, but everyone that I talk to about David says that he's a great collaborator, which is clear given the collaborative projects he's working on. He's always been helpful to me, and he does for a lot for the field, generally speaking, by supporting the careers of young scientists and promoting diversity and inclusion in academia, and being an advocate for the importance of animal research. He's really been a role model for and a hero for me and for many others in the field. I could go on, but I'll stop here to say thanks, David, Dr. Jentsch, for coming today. And take it away whenever you're ready.
Well, thank you very much, Paul. Can everyone hear me very well, please? All right. Well, with an introduction like that, I could let you go on and just defer the rest of the science. But in the interest of why we're all here together, I'll give you my science talk. Thanks everyone for taking time. I know even if this isn't the first week of classes for you like it is for me, this is a very difficult time of the year. And so I'm pleased that everyone was able to show up to hear what we've been working on over the past 15, 20 years or so, really. Because I'm going to try to give you a story that starts a number of years ago and ends up where we are today. So the focus of this talk is really going to be, as Paul mentioned, around my, my interests in impulsiveness and impulsive behaviors and their relevance for disorders of addiction. And I'm going to try to give you some insights into how genetics has really helped us to understand this phenomena better. All right. So, but before I move on, I just want to kind of get some, share my thinking about the phenomena that I'm going to talk most about today, just so we're all on the same page. I'm fundamentally interested in the motivational processes that encourage individuals to seek and consume rewards. A subset of those motivational urges could be referred to as impulses, the impulse to do something, a feeling state that drives one to desire to engage in behavior. And if those impulses lead to the expression of the behavior, they can result in impulsive behaviors. So impulses can drive impulsive behavior, except under circumstances where individuals expend some kind of effort to stop that process, to sever the relationship between the urge and the expression of the behavior. And it's that process, it's the process that allows us to inhibit or interrupt impulsive behaviors that really is sort of my cardinal interest in what I'm going to talk about a lot today. We all know that some people have a trait-like proclivity to give in to their impulsive urges and to engage in impulsive behaviors. We refer to that phenomena as impulsivity. And we think impulsivity could result either due to the fact that impulsive urges are exceedingly strong, or that those mechanisms that people use to inhibit or to interfere with impulsive behaviors are unusually weak. And so there's two sides to this coin, and I want you to be aware of that, sort of the drive and the stop mechanism. But a lot of my interest really has to do with the control mechanisms, the stop mechanisms. So if you need to understand this phenomena any better, you can just remember the age-old observation that individuals differ when they're presented with a relatively simple scenario. They're offered a scenario in which they are presented with a palatable reward, like a marshmallow, and they can either consume that reward now, or they can interfere with, stop their impulsive urge to consume the stimulus, because if they wait, they'll get a larger later reward, right? And so there's two things going on. There's some kind of, again, urgency that lends from the temptation to engage in the act of procuring and consuming the reward. But there's also this stop mechanism that says, if we can wait a little bit, things will be better for us. And I would contend that from a neuroscience perspective, we know actually a heck of a lot about why our brains like, want, and encourage our behavior to approach these palatable stimuli. And we know almost nothing about the flip side, which is why some individuals say, no, I'm prepared to wait. I'm prepared to delay gratification. And so that's really one of the things that's led me to be interested in this phenomena. And I would argue that the mechanisms we use to control our impulsive behaviors are broadly important to understanding well-being and healthy and unhealthy behaviors. And from the perspective of addiction in particular, individual differences in impulsivity at least partially predict the tendency of people to engage in various components of the life, essentially of the transition from recreational drug use into more hazardous and ultimately clinically impairing use, including elevated decision to initiate experience, the tendency to engage in more hazardous patterns of use, the transition from controlled to uncontrolled use, and ultimately difficulty with maintaining abstinence. This means that impulsivity and impulsive behaviors have pervasive interrelationships with the cycle of addiction. Not just addiction, right? Impulsivity, again, is a broadly important phenomena. Impulsivity features in other psychiatric conditions, as well as in other kinds of non-mental health disorders like overeating and other high-risk behaviors. So my interest in the relationship between addiction and impulsivity and inhibitory control actually have very early origins from when I was in graduate school. But after I took on my faculty position at UCLA, I began collaborating with Professor Edith London, who's on the faculty in psychiatry and biobehavioral sciences at UCLA. And of course, she runs a human neuroscience lab, and she had some of the same interests I did at the time in trying to understand these relationships between impulse control and addiction liability. And so we sought and ultimately attained funding from NIDA to support a developmental center, P20 center, whose goal was to understand this set of relationships between inhibitory control and addictions more deeply using a combination of animal and human approaches. And my research component involved examining how experience with a stimulant drug of abuse, in this case methamphetamine, because we received this funding in the early 2000s when that was an issue of predominant concern in the nation, would affect inhibitory control function, so the degree to which experience with the drug perturbed inhibitory control abilities. And I also was interested in the degree to which dopamine system signaling in the brain might mediate those relationships. And I'm going to show you some of the results from that. And this project was largely led by my research assistant and eventual graduate student at the time, Stephanie Groman, who's now an assistant professor in the Department of Pharmacology at the University of Minnesota and is running her own very exciting research program, which touches on very similar issues today. So what we did in this project is we were going to determine the causal effects of experience with methamphetamine on inhibitory control functions measured behaviorally, and as I'll show you next, on brain dopamine system function measured in vivo using positron emission tomography. And so we were going to, because of that, we were able to do a longitudinal study in which we assessed inhibitory control, dopamine system function before, as well as a short time after, as well as a long time after, a multi-week regimen exposure to escalating methamphetamine dosing. We used fervent monkeys, as you can see here, and our measure of inhibitory control was a reversal learning task. And so what we essentially do in this test is very simple. What we do in this procedure is we train subjects. Later, I'm going to show you that we have imported this procedure to mice. We train mice or monkeys, or indeed human subjects, that one response is associated with reward. For example, in this task, a discrimination task, monkeys learn that the box that has a red plus on the top has a reward inside of it. And so over trials, they become very, very quick and accurate at selecting the red plus box to obtain the reward that they want. But at some point, unexpectedly to the subject, the reinforcement contingency switches. And so responding to the red plus no longer procures reward. In fact, you have to respond to the gold circle. Subjects, it seems like a simple thing to do, but even subjects, even human subjects, even healthy human subjects struggle to some degree with the ability to stop the automated trained response, to select the red plus, and to update their behavior. And so at reversal, this stop and change component is linked with the ability to turn on this inhibitory control circuit and effectuate the interruption and that initially trained response and adapt behavior. And we have compelling evidence across species that the neural circuitry that's associated with this stop and change ability and reversal is highly conserved from mouse all the way to the healthy human subject. So we're using this reversal learning task. We're also combining this with measures of in vivo dopamine system function calculated with positron emission tomography to study, again, whether methamphetamine experience perturbs the inhibitory control abilities of subjects and what are the mechanisms by which that happens. So first, I'll show you the neurochemical results that we gained in the study here. Again, we have 3 time points on the x-axis before, a short time after, a long time after, a multi-week dosing regimen with methamphetamine. And what you're seeing on the left is a measure of presynaptic dopamine function. So we measured dopamine transporter availability. And then on the right, we measured postsynaptic D2-like receptor availability using F18 phthalipride. And what you find, just from a neurochemical perspective, is what we actually expected and thought would happen. That methamphetamine experience was sufficient to disrupt both pre and postsynaptic measures of dopamine system function. You can see here that in multiple parts of the basal ganglia, dopamine transporter availability declines, and it stays declined for a fairly long period of time after the drug exposure has ended. If you look at postsynaptic D2-like receptors, you find that drug experience was sufficient to reduce D2 receptor availability. Again, short time after methamphetamine, a long time after methamphetamine. And for those of you in the field, you know that these phenomena fully recapitulate what's observed in humans who have a methamphetamine use disorder, both evidence of presynaptic dopamine complications and pre and postsynaptic modifications in dopamine signaling. At the same time, we found what we hypothesized, which was that methamphetamine exposure was sufficient to perturb responding on this reversal learning test. It induced an inhibitory control problem that wasn't there to begin with. And statistically, behavior was well predicted by the decline in D2 receptor changes. The greater the D2 receptors, the greater the decline in D2 receptors the animal experienced, the greater the inhibitory control deficit they acquired as a function of drug experience. We don't think that they're just correlated. We think that they're causally linked with one another, because we actually already knew that if we pharmacologically interfered with D2 receptor signaling, so instead of causing the receptor abundance to decline because of methamphetamine, if we simply inhibited the activity of the receptors themselves that were there, that's sufficient to fully recapitulate the kind of inhibitory control problem that the other monkeys got because of the methamphetamine exposure. So we're pretty convinced that low D2 receptor activity, whether it's pharmacologically induced, whether it's a result of methamphetamine experience, is sufficient to create this state of a sort of deficient control over reward-guided responses. So what we learned in this study is that methamphetamine experience was sufficient to impair inhibitory control over impulsive behaviors. It compromised both pre and postsynaptic dopamine system function in the basal ganglia. And our conclusion was that reductions in brain dopamine D2 receptors caused by the drug probably negatively impacted in the circuitry that's responsible for inhibitory control and produced this association. Of course, we found some other interesting things while we were doing this study. And one of the things that's, I think as somebody who's worked in non-human primate models for many years, is never far from our minds, but I think is actually often far from the minds of rodent researchers, is that every one of the subjects is an individual. They're behaviorally and physically and in every way possible, unique individuals. And so inter-individual variation is quite great in our studies when we use non-human primates. And that certainly was true in this study as well. And in fact, when we looked at the data, in this case, the data that was gathered from subjects before the first methamphetamine dose was given at baseline, we measured at baseline their behavior. We measured their dopamine D2 receptors and their dopamine transporters. And you can see the result of an analysis of what's going on even before any experimental perturbation. You can see that down here in C. This is plotting D2 receptor availability. This is the native individual differences that they came to the study with on the x-axis versus their inhibitory control abilities on the y-axis. Some monkeys were really good at the task. Some were really poor. Some had low D2 receptors, some had high D2 receptors. And what I'm showing you was that the relationship between the two was exceedingly strong. In fact, individual differences in D2 receptor availability in the basal ganglia explained more than half of the variability in the behavior. So without any perturbation, experimental perturbation, there's already this really strong relationship between brain dopamine signaling and inhibitory control abilities. And yes, the drug can further perturb that, but as I'll say in the next slide, this effect, the individual difference effect, massively outweighed the drug-induced perturbation effect. This was where all the money was in this study, even though this was not what the study was designed to actually see. So yes, methamphetamine exposure, and we've studied other drugs of abuse as well, produces as an effect neuroadaptations that elicit inhibitory control deficits, and that adaptations in dopamine system function are at least a part of that. But again, the size of that effect pales in comparison to essentially nature's experiment, which configured individual differences in brain D2 receptor function prior to first drug exposure, and seems to associate very, very strongly with native differences in this cognitive ability. Subsequently to our study, the same relationship between individual differences in basal ganglia D2 receptors and inhibitory control abilities has been found in rodent subjects, in healthy human subjects. And so this appears to be, I would argue, probably one of the most reproducible biomarker biobehavioral phenotype relationships that's observed across species. And so this is really where my interest in individual differences came from. And I came away from this study thinking that I had never written a grant to propose to study individual differences, but this had to be a part of what I did in every study after this one. Individual differences really had to feature into our theories and experimental approaches in this field. And that the sources of those individual differences, which include but are not limited to diverse genomes, also include diverse life experiences, also including diverse developmental trajectories, which all sort of accumulate to affect individual difference in risk for addiction and for these other endophenotypes like inhibitory control, really had to be something we would study better in subsequent research. And so while we could have unpacked any of those contributors to individual differences in risk, and clearly we do, we also study how life experiences and developmental trajectories affect these phenomena. Genetic variation is just known to be a large contributor of risk for these phenomena. And so it was a tractable target to begin to sort of unpack subsequently. At this point, I'm really going to shift my research, the research I'm presenting, to the rodent model. We subsequently were very intrigued in being able to use non-human primate samples to understand individual differences at a genetic level better. Our animals all came from a pedigreed colony, which were sort of optimal for genome-wide linkage studies. But that research eventually started to peter out after our colony got transferred from UCLA to Wake Forest and then reduced in size and was no longer sort of suitable for genome scale studies. And that really caused us to pivot towards studying the mouse model. And for many people like me who had very little experience with mice at the time, the question was, what mouse resources would we use to further study this phenomena? Everybody in the study, most people in the field are studying one or a relatively small number of genome types that weren't selected because they were particularly remarkable for the trait of interest, but just because that's what people did. So I began to really try to seek input and advice and collaborations that would be kind of open the door for me able to use mouse resources to better understand this. It turns out that was not that hard for me. Jake Lucis, who developed the hybrid mouse diversity panel for use in genetic research, it was and is at UCLA. And so I went to his office and talked a lot about how we could use HMDP, which is essentially, for those of you who don't know, the HMDP is made up of hundreds of genetically unique inbred mouse strains that can be used to study sort of the heritability and coheritability of traits, and that can be used in sort of conventional systems genetic analysis. Jake had really developed a lot of the approaches that would make the HMDP highly suitable for a study of our type. And I did actually, our first research was kind of in this area. I was also, of course, very aware of the recombinant inbred mouse populations, which make up a large subset of the HMDP, I should say. They're not exclusive from one another's populations that could be used for many of the same purposes. I was sort of less aware and less confident in moving in the direction of using outbred subjects to undertake some of these research projects. But as I'll show you at the end, we were able to eventually move in that direction as well. And so over a number of years, we've actually taken our hypotheses, our approaches into these different mouse populations in order to better understand some of the genetic inputs for individual differences in inhibitory control and addiction liability. So this is showing you, I'm going to take you through a number of different sort of specific results that have come from our research using these different populations. So here's a study from the BXD recombinant inbred mouse panel. My graduate student at the time, Rick Laughlin, had actually already had was a student in the program who transferred into my lab. And before he came to my lab, he was already working with Rob Williams on using the BXDs for some neuroanatomical trait dissection studies, and actually published a paper with Rob prior to coming into my lab. And he had this idea that he could bring his experience with this system into our kind of complex behavioral phenotype space, which I thought was actually quite insane at the time. But I basically said, whatever you want to do, more power to you. And so Rick said about using the BXD panel to understand heritability and genetic determination and genetic influences on reversal learning performance in BXD mice. We developed an operant conditioning approach in which animals actually learn a spatial discrimination. They learn to respond to a particular hold to get a reward. And then unexpectedly, just like in the monkeys, the rule changes and the animal has to be able to adapt its behavior accordingly. And what you can see on the bottom, for those of you who are aficionados of graphs of this type, is the evidence that the BXD panel, here we assessed 51 strains, including the founder strains. And what you can see is evidence of a complexly determined and reasonably heritable trait. And so I'm not showing you the math for that, but I think most of you can look at that and see that's what you see that in this pattern of data. And what was sort of not really hypothesized by us at the time and ended up being a result we found after the fact is that after we completed this large strain survey of reversal learning performance in BXD mice, the main goal was to do genome-wide linkage analysis, but at the same time, of course, one also goes in search of genetic correlations using gene network resources. And when we uploaded all of our panel data to genenetwork.org and just said, tell us the top 50 genetic correlations for our trait, the second highest genetic correlation was for dopamine D2 receptor abundance in multiple brain regions of the brain. And so this really added to our observation in monkeys. It added to the observation that came from other labs in humans and in rats that suggested across all these species, by whatever mechanism you end up with low D2 receptor abundance, you end up with poor inhibitory control and vice versa. And what our result indeed also added to that is the proof that that was a genetic correlation that's producing that association between D2 receptors and inhibitory controllability. So though this was not necessarily a hypothesized aspect of the study, it ended up being one that really convinced us that, again, across species, this behavioral marker at least associates with, in a very similar pattern with molecular processes in a highly conserved fashion across at least across mammals, many mammals. So we then used our sort of conventional approaches to map quantitative trait loci for this trait in the BXD panel. And here you can see the chromosome 10 locus that emerged in our analyses. And we then used all the sort of conventional approaches that many of you use in order to identify candidates that were candidate genes that were expressed from this interval. The main approach we used is we tried to identify genes expressed from the interval that exhibited a cis-CQTL and whose gene dosage was correlated, genetically correlated with the level of expression of the behavioral trait. And so when we did this, we actually found 3 candidates expressed from this region that satisfy those criterion, meaning that they are cis-CQTLs and that expression trait phenotype is genetically correlated with the behavioral trait. And we also added the criterion that we had to be able to observe that in multiple tissues. One tissue was not sufficient in order to nominate a candidate. And so there were 3 genes expressed from this region that satisfied that criterion. One of them being SYN3, the gene SYN3 that encodes synapsin 3, which is actually really at the peak of the interval, which is not the reason to prioritize it. There's a number of reasons we were interested in this. This is the one that was most uniquely expressed in the brain, like all the synapsins. It's a brain-specific, a neuron-specific protein. And here on the bottom left, you can see the kind of evidence that led to its nomination. So SYN3 expression, for example, in this case, in the nucleus accumbens, is genetically correlated with the behavioral phenotype, and it's in the direction that underexpression of SYN3 appears to confer the poorest inhibitory control abilities. Now, we knew what synapsins were at the time, but we really didn't know anything about SYN3 in particular, where it was expressed in the brain and how its effects might be come to be, essentially. We know that SYN3, like all synapsins, are synaptic phosphoproteins encoded by 3 different genes that segregate vesicles away from the ready releasable pool. So when synapsin expression is high, when its activity is high, the ready releasable pool is low. You deplete the ready releasable pool, right? And so actually neural activity inactivates synapsin through phosphorylation so that you can increase neurotransmitter availability at the ready releasable pool. Synapsins 1 and 2 are essentially ubiquitous proteins. They're expressed throughout the brain. Probably many of you doing viral vector work know that we use synapsin 1 to produce pan-neuronal expression of our targets. And so if synapsin 3 had that same pattern of expression, we wouldn't know anything about its functionality. But SYN3 is actually quite different than SYN1 and SYN2. It is not expressed ubiquitously in the nervous system. If you go to the Allen Brain Atlas, you'll see that its expression is actually quite restricted in the brain. And one of the regions it is expressed at high density is in the ventral mesencephalic dopamine neurons of the substantia nigra and VTA, which has a potentially an interesting characteristic about it, given what we already knew about dopamine and its relationship to reversal learning. SYN3 is also different than SYN1 and SYN2. SYN1 and SYN2 are essentially fully redundant with one another. You get no phenotype in the SYN1 knockout or the SYN2 knockout. You make a double SYN1, SYN2 knockout, and the animals have all kinds of problems, mostly seizure disorders because you disrupt the GABA ready releasable pool. Not true of SYN3. And so basically about 6 months after we completed our linkage study and had SYN3 and our mouse as a candidate, this paper from Whiteman's lab came out in the Journal of Neuroscience. And so he also recognized that SYN3 was expressed in dopamine nerve terminals, and he set out to determine whether SYN3 controlled the dopamine ready releasable pool. And this is the study from this, the results from his study that confirmed that. So if you see, these are dopamine release events measured with voltammetry. In the top panel, you see a triple knockout, no SYN1, SYN2, SYN3. Dopamine releasable pool is much greater, which is what you'd expect because there's no SYN isoform to control that readily releasable pool. And then you see on the bottom panel what happens when you selectively delete SYN3. You fully recapitulate the effect of deletion of all 3 synapsins. So he was able to show that in fact SYN3 is the synapsin of isoform that controls the readily releasable pool for dopamine. And so all that together really helped us to think pretty confidently that SYN3 was likely to be our functional candidate at this locus that was controlling reversal learning, and then and also that effect was mediated by perturbations in essentially phasic dopamine release. And so we went to the Knockout Mouse Project, who had already created a SYN3 null allele. We acquired those mice and we utilized them in the same test we used to confirm or to conduct the BXD studies, and we confirmed that SYN3 deletion is sufficient to affect our reversal learning procedure in, I should say, in the same direction that our directional hypothesis came from. Because remember, it's underexpression of SYN3, that a heritable underexpression of SYN3 that conferred poor reversal learning, and that's exactly what happens when you delete the SYN3 allele. So genetic factors, including SYN3, are associated with reversal learning performance, and we think that their ability to as a mediating mechanism to regulate phasic dopamine release, at least in the case of SYN3, from the ready releasable pool, likely explains that relationship. And this will become interesting again in my very last data slide. So one of the reasons we study reversal learning is because we thought that it measured a cognitive construct that shares some relationship to addiction behaviors. And so having done this work with reversal learning and trying to figure out its genetic basis, it's important for us to kind of then bring that back or link that back with addiction, more addiction relevant traits like self-administration itself. And so we have set up a program in order to use intravenous self-administration in mice in sort of some overlapping genetic reference populations in order to understand these relationships better. Not shown in my presentation today, we knew that if we were able, for example, to go to BXD strains selected from the reference population because they had extreme phenotypes on the reversal learning test, we got predicted effects on self-administration. Those strains that were deficient in reversal learning had poor inhibitory control, self-administer larger amounts of the drug. And again, that shows that that association is a genetic correlation, since it was the same strains, but different mice. And so now we really feel that we can take these self-administration phenotypes and sort of further power in to understand the genetics of this trait. So a number of years ago, I got a U01 grant as part of this, the NIDA Animal Genetics Consortium, along with Des Smith at UCLA, to use the HMDP, which includes many of these BXD mouse panel strains that I've already been talking about, to better understand the genetic contributions to self-administration phenotypes. And so as of maybe a month and a half ago, we've completed intravenous cocaine self-administration testing on more than 950 mice from more than 80 inbred strains, including about half being BXD strains. It's a very simple procedure. The animals have 10 consecutive days of testing in which they're allowed to self-administer cocaine on a FR1 schedule of reinforcement. I should say this study is a little bit more complicated than that, and that half the subjects were allowed the opportunity to self-administer saline. You may look at me and say, why would you do that? That sounds insane. And the reason is because there's a differential expression component to this study. We wanted to study cocaine self-administration induced changes in gene expression as their own set of heritable traits. And to do that, we needed the saline condition. The animals get the same surgery. They get all the same testing. They just can't get access to any cocaine. Right. And so that's what makes up this particular study. And this study allows us to investigate essentially the heritability of cocaine self-administration traits, but also eventually differential expression of genes in the brain that may explain strain level variation in the behavior. And so this is the work mostly of my postdoc, Jared Bagley, and my research assistant, Barbara Force. And so what you're seeing here are cocaine self-administration phenotypes on a large number of inbred mouse backgrounds. This is, to make this simple, because there are 950 lines in this graph, this is the post-acquisition level of intake. So we do, I mean, it is a very straightforward, simple procedure, but by 7, 8, 9, 10 days of training, there's really no observable day-to-day change in the behavior. So we consider this to be post-acquisition responding. This is essentially this is the level of post-acquisition preference for cocaine, if it were, as it were. Okay, and so once again, you can see a data set that are consistent with a complexly determined relatively heritable traits. So here, heritability certainly exceeds 50% for this particular phenotype. I've also shown you data from male and female subjects. So all the animals in all the strains and all the conditions were half male and half female. And although it's difficult for you to see, I'm just going to point it out to you. There is no main effect of sex, not in this study, nor in any data set I'm going to show you subsequently. However, we consistently find sex by genotype interactions over and over and over and over. So there's no sex difference. There's a genetically moderated sex difference. In some genetic backgrounds, females take more cocaine than males. In others, there's no sex difference. In others, males take more cocaine than females. And I think this is an underappreciated thing, certainly outside of a group like this, that we really need to pay attention to these genotype by sex interactions, because they're really what is happening in large diverse populations, not simple sex differences. In addition, I'll just point out C57 Black 6J here, which again, in this population shows a pretty unremarkable phenotype. They self-administer the drug. They don't self-administer the drug, they're in the lower 50% of the distribution for this trait. You can see that with the purple arrow here. And one of the things we've kind of learned along the way of doing these studies is the importance of doing these population studies so that we can actually identify the strains that are the really interesting ones that should be the target, the subjects in our research, right? So again, there's a large number of BXD strains in the HMDP. About half the strains are BXDs. And so you can see the DBA2J here is actually the lowest responding subject. You can again see the C57 Black 6J, which is in the lower 50% of the distribution. But then you can see on the far right that BXDs disproportionately make up the highest responding animals in this subject. And we are now arguing that these are really exciting strains to study. They're polygenic models of extreme cocaine-taking behavior by virtue of the fact that they, got unlucky in the draw of alleles from the ancestors, and they got all the bad ones. And these really are the animals that we should be studying in our I think we should be studying in our neurobiological studies, where we think we're studying addiction-like phenomena. So we can use these large HMDP populations to do genome-wide associations. This is sort of a Manhattan plot for, again, the post-acquisition level of cocaine preference. And we have a strong QTL on mouse chromosome 14 and a sort of a weaker one on mouse chromosome 3. And we're going to use all the same approaches to try to understand positional candidates that are expressed from this QTL that might be responsible for the trait. And so, as I said, this study was designed to be a gene expression study. All the animals were sacrificed 24 hours after their 10th day of either saline or cocaine self-administration. And medial frontal cortex and ventral striatum were collected, and we performed, with DES, we performed RNA sequencing on all these samples. And here you're seeing gene expression correlates to behavior at the strain level, either in animals who had a saline history. So this means if there's a gene expression behavior correlation, it's solely due to genetic effects, right? Because these animals have never had any cocaine experience. We also have potential relationships between the behavior and gene expression in the cocaine experienced subjects. And then we also calculated the differential expression. So cocaine minus saline, cocaine induced change. And what you can see is that from the chromosome 14 locus, there are genes whose expression levels, either in the cocaine experienced animals, in the difference score, or in the baseline, essentially the heritable, where only heritability is at play, you observe correlations to the behavior. And so we think that this approach is sort of getting us closer to understanding genes that are likely responsible for essentially the individual differences and the tendency to take cocaine in this self-administration test. So of course, these populations are genetically and phenotypically variable, but they're still themselves not as phenotypically or genetically diverse as we are, or as are the monkeys in my earlier study. And so it was that recognition that really led us to begin collaborating with the Chesler Lab and others at Jackson Laboratory to study these phenomena in even more diverse genome types. And so here we're talking about the collaborative cross and diversity outbred mice that resulted from the intercross of these 8 inbred founders. Everybody knows the rationale for using these populations. They maximize the amount of genetic diversity and allelic variation in the population so that we could presumably find more variants that are associated with our trait. And so we started a project, NIDA-funded P50 Center with Alyssa Chesler, Lisa Tarantino, and Colleen McClung at Pittsburgh about 5 years ago now. And my project proposed to use the founders, the collaborative cross and diversity outcross mice to study this reverse learning trait, kind of in the ways we've done in other populations, but in this more genetically diverse population. We've used them to study a number of different traits. I'm going to show you the reversal learning test here, but this is just to sort of support the idea that the concept behind using these more genetically diverse panels actually pans out. So here we looked at them, the founders and a subset of CC strains for their consumption of and preference for an ethanol solution, oral ethanol solution, which everybody knows the C57 black 6J mice are supposed to be the most robust drinking strains that people observe. But here you can see the PWK is one of the wild-derived founder strains that were used to generate the collaborative cross mice. They show, double the levels of ethanol intake and much higher levels of preference than our canonical black 6J strain does. And that sort of shows that the expectation that greater genetic diversity would confer greater phenotypic diversity does actually pan out, at least in some circumstances in this one. So we've studied reversal learning both in the founders shown here, as well as a large and growing number of CC strains, which are recombinant inbred strains derived from this intercross. And once again, you can see data that comports the idea that this is a complex and heritable trait. And further supports the idea that, again, black 6J mice are probably not the ideal mice to study if you want to study the neural mechanisms of high impulsivity. They're in the top 50% by this token, but certainly not anywhere close to the top CC strains. So we've completed reversal learning testing in almost 400 diversity outbred mice by now. Of all the phenotypes we studied in the center, the reversal learning phenotype readily emerged as one that has a very robust behavioral QTL here on mouse chromosome 7, which meets the p less than 0.01 FDR threshold for genome-wide significance. This interval still expresses multiple genes, of course. We're not going to get to single gene level resolution with any of these approaches. But once again, I'll show you in the next slide, we've used all the conventional approaches, which include identifying genes that harbor non-synonymous coding variants, and as well as identifying genes who are cis-regulated, have cis-eQTLs, and whose expression is genetically correlated with our trait to nominate positional candidates. And that effort led to the identification of a gene called WDR73, which is the W domain repeat containing gene 73. It's part of a large family of genes called WDR. What's strange about this family is they have nothing functionally in common. They just all have repeats, and so they got called the same thing. And we actually know relatively little about WDR73. All I can really tell you is that in humans anyway, haploinsufficiency in WDR73 is associated with phenotypes, and they are all neurological. All the phenotypes associated with WDR73 insufficiency are brain phenotypes. But after that, the information drops off pretty precipitously. Of course, that's the risk and the benefit of this kind of approach, is that you find things that you then have to spend a hell of a lot of time trying to figure out the mechanisms that they might act upon to exert these effects. WDR73 exhibit, again, it is cis-regulated, has a cis-eQTL, and its gene expression dosage in the CC mice is genetically correlated with reversal learning performance. It is in the positive direction, so strains that overexpress WDR73 exhibit the poorest inhibitory control function, and vice versa. And so we're currently have funding proposals under review to sort of further examine WDR73 functionality and understand this effect better. Although there was no reason, it's very little known about WDR73, and there's very little reason to have selected it as a candidate based upon its known function. Once again, exploratory phenomics analysis help us to think that we might be on the right track. So we took WDR73 and we went to systemsgenetic.org and we asked the question, which phenotypes in genenetwork.org in BXD mice are genetically correlated with expression of WDR73 in the brain, if you specifically focus on the brain regions we're interested in, right, medial frontal cortex and ventral striatum. And what you see is the results of that ePHEWAS here. So 4 phenotypes met phenome-wide significance, exceeded the phenome-wide significant threshold for significance. And those traits are dopamine receptors, D1, D2 receptor ratio, D2 receptor expression, D1 receptor expression. And so although there's really essentially no reason to have posited in advance that WDR73 was associated with reversal learning, the exploratory analysis said that it likely was. And even though there's no reason to believe its effects would be mediated by actions on dopamine, the ePHEWAS say that that's likely to be the case. Now, how that effect plays out, I cannot tell you today. I do not know. This is as far as we know about this set of associations and the potential mediating effects of dopamine receptors on this set of phenotypes. So in closing, I think we can, by harnessing the diverse mouse genome, we can essentially definitively identify mechanisms that regulate phasic dopamine release, D2 receptor functionality, and consistently are associated with this inhibitory controllability. In the process, I think we can discover and harness better polygenic models for addiction risk so that we can do better research into the neurobiology of underlying the disposition for addictions. We can localize these genetic regulators and begin the process of identifying how they affect behavior through our known addiction circuits. And we can really find molecules and pathways that are markers for addiction risk, and really importantly are potentially entry points for severing the genotype-phenotype relationship. So in closing, I want to acknowledge my faculty collaborators at UCLA, Des Smith and Edith London, as well as my trainees, Stephanie Groman and Rick Laughlin at UCLA, who participated in these studies. My current lab staff, Jared Bagley, Lauren Bailey, and Alyssa Moore, who performed various parts of these studies, and my collaborators in the systems for the Center for Systems and Neurogenetics of Addictions, Alyssa Chesler, Alyssa Tarantino, Vivek Phillip, and Leona Gagnon. And so with that, I think I am done, and we have some time left over for questions.
Thanks, David. That was excellent. I see clapping all around in the participant list as well. Thanks again for giving this talk. There's a couple of questions in the chat that I'll read. And then after that, either people can raise their hands on the participant list and I'll call on them. But if there aren't a lot of hands raised, then we can just sort of feel free to unmute ourselves and ask them freely. So I'll start with a question from Ankush Chakraborty. He asks, did you try to do a longitudinal study to see if due to environmental factors there can be epigenetic changes in the amount of dopamine receptors or something related to SIM3?
That, of course, is an excellent question, since we know there are a number of environmental factors, as well as I want to reinforce developmental trajectories that also regulate risk for addictions, and that regulate inhibitory control capacities and so forth. There's also, right, these things are known. The answer is we haven't studied that. That would be a logical thing to do as we try to integrate and better support or develop better support for the overall hypothesis that these neural mechanisms are sort of bottlenecks or convergence points for the multiple risk factors that regulate the same behavioral endpoints. And so that would be something, it would be absolutely be of interest to look at in the future. And we're just not there yet. I mean, in all fairness, the genetic experiment was seemingly the easiest to do from the beginning, right? I mean, so that was the logical starting point for us, but these other components are clearly worth doing as well.
Thank you. Before I get to the hands, one more in the chat from Megan Mulligan. Did you identify or prioritize functional segregating SIN3 sequence variants in the BXDs?
Well, of course, that would have been the next step in the SIN3 line of study. The answer was, in short, it's no. We essentially moved away from that set of associations as we were sort of drawn into the DO population, and we just didn't explore it deeply enough. That's a logical question. I will tell you that we have done that in the DO. And so there, for example, from my DO result, the WDR73 expression relationship to the behavioral phenotype seems to be explained by a structural variant within the WDR73 gene. And so we actually got really lucky that that particular SV came out as being, quite interesting. It also shows that SV explains the predicted founder effects on the, on the phenotype. And so we think in the case of WDR73, we actually are the answer would, if you said WDR73, I could have said yes, we think we have that variant, but not for SIN3.
Okay, I will call on Ankush and then Dr. Grisel. Go ahead, Ankush.
Hi, can you hear me?
I can, yes.
So I wanted to ask, this is probably a silly question, but then I wanted to ask, is there any evidence that saline can be addictive?
So is there any evidence that, the only word I missed was that one, that something can be addictive?
Saline.
Saline, oh, saline infusions. So to answer your question, we found across our population that there were a substantial number of strains that did respond for the saline reinforcer, okay? There was a modest correlation between responding for the saline reinforcer and the cocaine reinforcer. So I should just back up. cocaine is a much more effective reinforcer than saline. None of us are surprised by that, okay? There is a modest level correlation between responding for the saline reinforcer and the cocaine reinforcer. We don't know why. It's not probably because saline is itself a reinforcer. It's that there were other environmental events that were associated with the infusion. So like in most studies, when the pump turned on, there was also a flashing light that told the animal that a reinforcer was being delivered. So we don't know, if, for example, in that case, whether the animal enjoys seeing flashing lights. There's certainly evidence that sensory events like lights can act as operant reinforcers for operant behavior. Some people have studied that from the context of like stimulation seeking or sensation seeking. So there's evidence that it can be a reinforcer. That, of course, that's a far cry from saying that saline is addictive. We don't know that. We wouldn't immediately comport the fact that an animal will self-administer a stimulus with saying that that stimulus is addictive, or that behavior is one of addiction. But yes, something about the environmental test that we did, despite having cocaine not be in the infusion, there was something modestly reinforcing about that for some genetic backgrounds. And let's be clear about that. This is another, again, genetically informed trait, because there are other strains that who couldn't give to you know what about saline and the light stimulus. So it's itself its own kind of heritable phenomena.
And interestingly, it does sort of support the idea that, you know, one of the things that cocaine might be doing broadly is sort of energizing behaviors that animals are predisposed to be doing, you know, in the absence of cocaine. And I think that the locomotor sort of response studies suggest that too, because there's correlation between saline and cocaine-induced locomotion as well.
Yes, you're right. Absolutely. Judy?
Yeah, thank you so much. That was a fantastic talk. I loved it. And I just want to follow up a little bit on that magnificent sex difference you had in some strains and not others, especially the, I don't know how many fold change in that 129. So I wonder if you've looked at D2 expression in any of the pairs, you know, male and female pairs in those kinds of strains, or any of your other gene links.
No, the short answer is no. There is evidence along those lines. So Edith London has published studies on sex differences in D2 receptor abundance and availability in human subjects, including human subjects who have a substance use disorder. And she does find differences in D2 receptors that essentially comport with our hypothesized directional changes. But you're right that it would be of interest to study this in the context of say strains that maximally exhibit different kinds of sex differences from one another. So we do know, we have kind of, I'm pulling back from your question, which was really dopamine centered and really kind of opening it up and asking about mechanism more broadly. I become interested in whether across strains that shows different kinds of sex differences, whether that could be due to reorganization of the effect of sex biasing factors on those phenotypes. Like maybe of all the at least the three major sex biasing factors: sex chromosome complement, organizational effects of sex steroids, activational effects of sex steroids. Maybe they're actually maybe genetic background reshuffles them over and over again. And so to answer that question, we started with our alcohol consumption phenotype. So in the PWKs, we have remarkably higher levels of ethanol consumption than in the black 6J mice. But the sex difference in PWK PHJs is qualitative. I mean, there's no overlap between female and male drinking. The females drink more than twice the levels of alcohol than the males do. The sex difference in black 6J mice is much more subtle than that. So what we did to try to address this question of, are sex differences being are sex biasing inputs being reorganized? We took a mouse that's called the 4-core genotype mouse. And so this mouse model works because there's a targeted disruption of the endogenous SRY locus. And when you render the SRY locus null, you get an XY mouse who is gonadally and reproductively female. And then SRY was introduced onto an autosome. So it now segregates independently of the sex chromosomes, and that produces a mouse that's XX that has testes and is reproductively male. And so this has been on the black 6J background for a long period of time. It's been studied a lot. And of course, you can then determine what are the inputs, the sex splicing factors that contribute to sex differences? And we have really compelling data that the sex chromosomes are where the majority of the traction is in the black 6J background for producing sex differences. That after you break the relationship between sex chromosomes and gonads, the gonads just disappear as being an effector in pubertal and peri and post-pubertal mice. Then what we did is we made an F1 cross between the black 6J FCG mouse and the PWK PHJ strain. So these animals now have 50% genome type that is PWK PHJ and should predispose them for high drinking, which it does. But we can still produce the 4-core line on this B6J PWK F1. And when you do that, all the sex difference is gonadal. There's no sex chromosome complement effect any longer. So this tells me that genetic background is reshuffling the way that sex biasing factors change the behavior. Organizational, activational, genetic, and it really means that this, that kind of, to me, explains where these genotype by sex interactions come from. And so I would say that whether you're talking about those mechanisms, whether you're talking about dopamine system mediation, we really need to be able to study this more deeply. So we have started now doing, and we're proposing to do as part of the renewal center of our center, if it gets selected, to use ex vivo fast scan cyclic voltammetry across these strains to begin understanding that mechanism of how genotype affects behavior, possibly through regulating the mechanisms of dopamine transmission. And we have sort of pretty compelling data that the hypothesis is likely true, that is the case, that strain differences, when we select strains that are extreme according to the behavioral trait and we do FSCV on them, we find pretty consistent strain differences that are in the direction we would predict. So I think it's an important question, and I can't answer it fully, but I think you're probably likely, if the experiment was done, there would be reason to believe that you would find what you're predicting.
Thank you for that question, Judy. And thanks,
Sounds
David.
like it could be another talk he gives.
sounds like it. We are past 1 o'clock, but are there other questions? I just, maybe I can take a prerogative and just ask one last question. I'm just sort of like curious about your attitude towards candidate genes. When you found out about WR73, I fully agree with you wholeheartedly. It's like you have a gene, you don't know what the function is. That's actually kind of exciting. But then what's your reaction when you sort of zero into the idea that this might be mediated by dopamine receptors? Like, do you get excited about that being a proof of principle, something that you already know about? Or are you like, oh, this isn't, this is going to be, have a similar sort of story as the D2 story?
Yeah, so thank you for that question. I get excited about it. I mean, I think we have more certainty about what addiction circuitry is than we have about addiction genetics.
Yeah.
I think we know the things that really 40 years of research have said are the brain systems that at least in part contribute to the expression of these behaviors, right? And so while we shouldn't reject things that are outside of that scope, I think that understanding that at least a subset of the genetic associations to behavior are likely explained by the fact that the circuits that we know are involved is not a disqualifying factor. Some reviewers don't agree. Some reviewers don't agree. And so I think as long as we don't disqualify, like, would we throw out a gene who has a strong association but doesn't seem to do anything to dopamine? No, clearly not.
Right.
That would be a mistake. But I think as part of the overall evidence that the gene should be considered further, I think that's meritorious evidence.
That's great, and I wholeheartedly agree. The reason why I asked that is I wanted to steal some language from you that I can put in my next grant. So Ankush, you have your hand up. Let's make this the last question, and then we'll end it.
So I wanted to ask, do you know about if SEN3 affects addiction rates across different kinds of drugs, say cocaine, alcohol, and different drugs? Given
That's
that
a
it
great
is
question.
also, yeah.
I don't have a definitive answer. I know that as these mice have been profiled in the comp, which is why we were able to get access to these mice. And at least a sub-channel of the comp has looked at methamphetamine, oral methamphetamine self-administration. And I don't know I haven't seen the data, but I've been told that the animals do have a phenotype in the oral methamphetamine self-administration test. So that might say yes. I thought you were going to ask me a better, not a better question, sorry, a harder question, which is there any relationship between genetic variation in the SYN3 locus and addiction behaviors in people? And that is like an excellent question, but unfortunately, it's very difficult to answer. The SYN3 gene spans like 350,000 bases. It's a very, very, very large gene. And so even if you were to only fully genotype, even if you were to focus only on SYN3 and fully measure its genetic variation in a human sample, you need just tons and tons and tons of SNP markers to do that. So we've talked about that, but every time we end up deciding that it's just too much of a challenge to actually do. So there are actually, SYN3 has been a candidate study in human neuropsychiatric genetics for a long time. But essentially, despite being this enormous gene, everybody's genotyped it with one SNP and called it a day. And so it's essentially it's not fully tested.
Great. People are dropping off and we're past the hour. I want to be respectful of everyone's time. David, that was so good. Thank you so much. It was a great talk. You have an impressive amount of work that you've shown us today, and I know that's probably just the tip of the iceberg. As Judy mentioned, you have had a whole nother talk in the answer to her question. Anyways, I will say goodbye to everyone. Thanks again, David.
Thanks for inviting me.
And
Thank you.
we'll see everyone next month.
It was a wonderful presentation. Great data.
Yeah, thank you so much.
You guys are really.
Thank you so much.
Bye-bye.
Bye.
Oops. I want to tell Paul something. Let me see. And